Hello and welcome to Stories of Science. Here, we look back on humanity's long journey to understand the world. The questions, the failures, and the breakthroughs that slowly changed how we see the universe and ourselves. We tell science as stories.
The woman who unlocked life's code, and history almost ignored her. 1953. A black and white photograph. No filters. No fancy framing. A little blurry, even. But for the first time in history, humans saw themselves. This wasn't just a picture. It was the blueprint of life. Later, it would be called Photo 51. The woman behind the lens? A quiet scientist, she didn't like the spotlight. She wasn't the type to elbow her way forward. She did one thing: make the invisible visible. By then, people knew DNA existed, but what it looked like? Nobody had a clue. It was like owning a manual, but the cover was ripped off and the pages locked. And this photo, it was a key. A revelation. DNA wasn't a tangled mess. It had structure, grace. A twist, a helix, like a staircase, like a ladder, like life itself climbing upward. Beautiful, isn't it? But here's the catch: the photo changed the world. It didn't change her fate. History wrote down the discovery of DNA. It wrote two names in bold, not hers. Her name appeared somewhere else in the footnotes. If you open a textbook today, chances are you'll remember them, not her. It's like snapping the perfect photo. Someone else submits it to the contest, wins the prize, and it sits in your personal album. This photo was worth more than any selfie. She just didn't post it online. If Instagram existed back then, maybe history would have been different. But history doesn't do what ifs. So who was she, what did she do, and why didn't she get the most famous award in the world? That's where our story begins. Some people are born with talent. She was different. She relied on something trickier: grit, the kind of relentless, unreasonable grit. 1920. She was born in London. A classic elite family. Money, culture, clear expectations. You must excel. You must be useful. Sounds motivational, right? Except their definition of useful was narrow. Her father wanted her to do something respectable and safe. Teaching, social work, science, for a girl back then, way too weird. But she was weird from day one. She didn't like playing the games girls should play. She liked asking questions. Why does this happen? What if it didn't? Once she started, she couldn't stop. At school, she was consistent, not flashy, just reliable. Other kids had highs and lows. She didn't even know the concept of off day. Teachers said calm, rational, hard to deal with. Translation: not easily fooled. By 13, she'd made a decision: she would be a scientist. Back then, that wasn't just a choice. It was a declaration. Her father wasn't thrilled, not for lack of love, because reality was harsh. It was a time when women could study, but their degrees weren't recognized. It's like finishing an entire video game. The system says achievement not unlocked. She didn't care. Her logic was simple: if a problem exists, someone should solve it. She wasn't trying to change the world, she just wanted the eyesore of a problem removed. She got into top schools, pursued science. So the road wasn't easy, institutional roadblocks, human ones too. But one thing she didn't back down, not from problems. Argue with her, she'd show the data. If there wasn't enough, she'd run more experiments until the conclusion held up. Her hobby, proving others wrong. Sounds dangerous, but in science, it's called progress. While some kids rebelled with rock and roll, she rebelled with physics, deeper and deeper. She wasn't a lightning bolt genius, she was a machine: precise, tireless, unflinching. Once she locked onto a problem, that problem had no peace. She got into Cambridge. Sounds like a win, right? But soon she realized formal recognition? Not quite. 1938. Newnham College, University of Cambridge. A college for women sounds nice, but really, it was segregation. Rules were clear: women could attend classes, sit exams, excel, but could not get degrees. She did all the work. The system said achievement not unlocked. It's like running a marathon, crossing the finish line, and the judge says you did great, but it doesn't count. Most would be discouraged, not her. She kept going, not for the system, for the problem. That moment left a mark. She realized the world doesn't reward ability, sometimes just rules, and rules ain't always fair. Her approach to research carried that rebellion. She trusted data, not authority, because people lie. Data doesn't. Then a bigger reality hit, war. 1939. World War II broke out. Europe was on high alert as science became a tool. Some worked on weapons. Explosions. She chose something less glamorous, coal. Yes, coal. Sounds old-fashioned, doesn't it? Boiler rooms. Black smoke. Low tech. But she wasn't seeing coal, she saw structure. Why does some coal burn better? Why does some barely light? The answer: pores. Coal isn't solid. It's like a sponge. At tiny spaces determine gas flow, oxygen intake, and combustion. She used X-rays, analytical techniques, built models, explained behavior. Sounds academic. But the application? Direct, more efficient combustion. Better coke is stable energy output. During wartime, that meant resources lasted longer, industry ran smoother, nations stood stronger. While others studied bombs, she studied how to make fire work smarter. Not glamorous, but critical. This work shaped her skill, finding crucial structure in the ordinary. A skill that would soon be used on something far more astonishing: life itself. She was boxed in back in Britain, but not for long. 1947. She left for Paris. A whole new lab. The air itself felt different, fewer rules, fewer you shouldn'ts. Here, results mattered, nothing else. For the first time, she felt it. Science could be free, not something you begged permission to do. And here, she learned a key skill: X-ray diffraction. Sounds fancy, right? The logic is actually simple. You can't see molecules, then hit them with X-rays. The rays bend, scatter, leave a pattern. The pattern, like a shadow. From that shadow, you work backward, reconstruct the structure. Easy to say, hard as nails to do. You need precision, patience. A little bit of stubborn obsession. She had all three. Where others saw blurry spots and shrugged, she zoomed in, measured again, calibrated again, until the data told the truth. She didn't guess, she knew. In those Paris years, she didn't just learn the technique, she mastered it. It wasn't X-rays she was using, she was taming them. Paris had more than just coffee. It had a microscope that could change a life. When she left, she was no longer the student stuck in bureaucracy, she was a fully fledged structural scientist. And fate had a bigger question waiting, more complex, more dangerous. DNA. 1951. Back in London. King's College. A team working on DNA. It sounded like a promotion, but complications came fast. Enter Maurice Wilkins, physicist, DNA researcher. The problem, from day one, the vibe was off. Wilkins assumed she was his assistant. She wasn't. She was an independent researcher. Translation: one thought he was the boss, the other thought she didn't need one. It was a combustible mix. No fights, but no real collaboration, little communication. Less understanding. The air, not hostile, but not exactly friendly either. And the environment, brutally unwelcoming to a female scientist. In meetings, she was ignored, her ideas challenged. Interrupted. Not occasional. Standard practice. Her response: classic her. She didn't argue over status, she let the data do the talking. To disagree? Fine. She'd run more experiments, let the evidence speak. But science isn't just data. It's people. Communication. Team rhythm. They had none. Of their problem wasn't science. It was ego. Meanwhile, the DNA secret edged closer, and she was standing at the epicenter. When she snapped that photo, she was one step from the answer. 1952. Quiet lab, machines humming. Her. An X-ray film developing. The image that would become Photo 51. It looked ordinary, blurry dots. Light, snow, static on a TV. But if you knew how to read it, it spoke. A clear X shape in the middle, not random. Structured. A pattern. A helix. She didn't guess, she calculated. Step by step, measured angles. Analyzed spacing. It computed repetition cycles. Slow. Precise. She confirmed the molecules were orderly, repeating patterns. Symmetry. DNA wasn't a tangled mess. It was unnervingly precise. She was on the verge of the final judgment, the helix. One final step to piece it all together, but she had a rule: no approximations, as she wanted certainty. She kept validating, calculating, double-checking. So close to the finish line. Meanwhile, on the other side, people didn't wait, they raced straight to the goal, some might even say they drove there. 1953. The answer was out. The world shook. DNA revealed double helix. Elegant. Symmetric. Perfect. Presented by two men, James Watson, Francis Crick, Cambridge researchers. They didn't run the tricky experiments, they built a model, used existing data, assembled the structure. Smart. Effective. The twist, that existing data, her fingerprints were all over it, the photo. The parameters. The clues. Her name. Not on the title, just in the background. She supplied the puzzle pieces, they painted the picture. That's science. And reality is some explore. Some express. History often remembers the latter first. It sparked controversy. Was the data used with permission? No simple answers. But one thing was clear: science isn't just discovery. It's people. Relationships. Timing is. She didn't lose on ability, she lost on insisting on absolute certainty. Others spoke first. She left DNA behind, but not science. Post 1953, she made a choice: exit King's College, not defeat. A new map. Birkbeck College, a new lab, new problems. This time, she didn't study how life exists. She studied how life breaks. Viruses. Dark? Maybe. Critical? Absolutely. Viruses are simple, almost not alive. No fluff. Pure, compressed code. She saw opportunity to understand virus structure, understand life's core rules. She focused on a classic subject: tobacco mosaic virus. TMV makes plants sick. Her tools: familiar, X-rays, diffraction, analysis. She treated the virus like a geometry problem, not emotion, not fear. Structure. Her discovery: viruses aren't random piles. They're ordered, like a machine. Most crucially, the RNA isn't outside. It's inside, protected by structure. This was huge. Genetic information isn't haphazard. It has position, logic. She quickly gained influence in the field: papers, data, conclusions, point one after another. She didn't look back at DNA, not because it didn't matter, because she had moved forward. It wasn't an ending, just a new chapter. Others debated who discovered what. She was already solving the next puzzle. While they studied life, she studied how to break it. Sounds villainous, actually, she was studying the rules, and rules are the true heart of science. This time, though, time itself was an opponent. 1956. She sensed something wrong: medical tests, ovarian cancer. A cold term. Heavy reality. Cause. Hard to pin down. Point one possibility: long-term X-ray exposure, the very tool she used to see the world, slowly harming her body. Irony, isn't it? She used light to understand structure. Light was destroying her. She knew the risk as she didn't stop. She cut back a little, but never left the lab, kept writing papers, mentoring students, analyzing data. Slower, yes, but still moving and negotiating with time. Give me a little longer. Time didn't answer. It just ticked on. She endured surgery, recovery, back to work. No complaints. No dramatics, she just did what could still be done. In the lab, she was still the quietest, still the most focused, only now, she knew, time wasn't on her side. She didn't have time to argue. But she left results. Clear results. Now, let's slow down. Look at what she did. One discovery at a time. Start with the famous one. DNA. Most people remember the model. The double helix. Elegant. Perfect, but a model doesn't appear from thin air. It needs data, and she pushed that data to the absolute limit. She saw two states of DNA. A and B. Sounds like letters? It's more than that. Point two completely different arrangements. Humidity changes the structure. Get it wrong, and the model fails. She measured a key number 2. Repeating distance. About 34 angstroms, not random. Calculated from diffraction patterns. And one more insight: the phosphate backbone is on the outside. Technical, yes, but crucial. It decides inside from outside, like building a house. Walls outside, walls inside. Mix it up, and the house collapses. Then there's the photo. The famous photo 51. A clear X shape, not guesswork. Evidence. Speaking for itself. She didn't announce the answer, she wrote it on the blackboard. Someone just needed to read it. Her work was the foundation. Invisible, but essential. Next, coal. Sounds boring, Old-fashioned. Maybe, but what she did was cutting edge. She turned black lumps into structured data. She found pores inside complex networks. Pores decide how gas enters, how it spreads, how it burns. Industry changed. No longer burn more. Now, burn smarter. Fuel, materials all improved. High-performance carbon materials today? Her work is their ancestor. Then, viruses. Late in her career. The most crucial, she studied shape, not symptoms. Not transmission. Structure itself. X-ray diffraction. Tobacco mosaic virus. Order, not chaos. A precise tube. RNA safely inside. Genetic information is protected, not scattered. This idea became core to structural biology. Proteins, viruses, big molecules, all understood this way. Her contribution? Not one breakthrough. A method. A lens. See the world through structure, quiet, precise, hard to ignore. Where did it go? Not stuck in labs. Into human life. Into your life. DNA structure solved. A huge door opened to before life exists. But rules unknown after, code readable. Genetics no longer mystical. Why you look like you do. Why you get sick. Answers appear. Medicine changed. Not just treating, predicting. Gene tests. Doctors can warn you. Future risks scary? Maybe. Useful? Absolutely, like checking weather before leaving home. Then biotech. Humans rewriting code. CRISPR. Gene editing. Sci-fi? Reality, altered genes. Treat diseases. Change traits. All starts with, what does DNA look like? No understanding, no fixing. She gave us the manual. DNA tests today? In a way, she wrote the first page. She never stood in the spotlight, but she turned the lights on. Nobel Prize? She didn't get it, but she changed the post-Nobel world. 1962.3 people on stage. Watson, Crick, Wilkins, applause. Bright lights. History seemed settled. One name missing. Rosalind Franklin, she died in 1958. Age 37. Nobel rules don't award the deceased. Fate, not just rules. If she had lived, would she stand there? Some say yes. Some say maybe. Is science? Partly. Human nature? Definitely. Here's the uncomfortable truth. Her key data. Photo 51. Didn't walk to Watson. Taken, without clear permission, like your paper being published before you. A familiar? Gray area. Back then, common. Science was high stakes. Rules on paper. Street rules in play. Her story rang a bell. Who owns data? The lab? The person? Whoever uses it first. Once asked, can't unask. System changed. Author lists grew. Credit precise, not for show. For protection. Women in science? Labs tolerated them, quietly. Her rules, be there, but don't stand out. She broke them all. Logical. Exacting. No fuzzy talk. Pure scientist. Critics called her difficult. Too assertive. A swap gender. Same traits praised. Not her fault. Environment's fault. She didn't argue. She experimented. Better experiments. Others noticed. Slowly, more women entered science. Normal now, not exceptions. Cornerstones. Back to the stage. If she lived? Watson excited. Crick meticulous. She, quiet, in the corner. Double helix? Not inspiration. Data, coaxed out slowly. Some say science is lightning strikes. She'd smile. Disagree. Her world. No miracles. Precision only. Angles measured. Spacing calculated. Symmetry analyzed. Each number a nail securing truth. Others saw structure. She had seen evidence. If alive, she might stand on the podium. But more importantly, she'd keep working, not chasing fame. Already chasing the next question. Viruses, RNA, unknown forms, awards? Side effect. Her life. Not about prizes. Right versus wrong. Not who speaks first. Reality? Complicated. Regret. Maybe guilt. But change happened. Modern science emphasizes transparency, fairness, respect, not because people got kinder. Because history taught lessons. History doesn't repeat, but it rhymes. Her story? The clearest rhyme. She didn't win the race, but she changed the rules. Harder than winning once. Changing rules, everyone agrees, today? More and more do. When you see DNA, think Watson, Crick. Also think Rosalind Franklin, the one at the microscope. The one who didn't argue. Just measured. She let the world see structure. Clear. No drama. No memoirs. Just data. Cool. Precise. Irrefutable. That data became the foundation for understanding life. Your DNA test? You might like coffee report? She wrote that first page, exaggerated? Not at all. From the double helix onward, humans understood heredity from lab to hospital to daily life. Drugs, diagnostics, biotech, all flows from there. She stood at the start. No applause, no lights, just X-ray beams. One photograph. History is noisy. Awards, controversy, names flying. The truly important stuff? Quiet.
The people who change the world usually just think one step deeper. If this story made you see things differently, hit like, subscribe, and save it. See you in the next story.